In details, we develop a new gold standard for the screening of breast cancer and prostate cancer. The goal of Eucrasyc is to create an easy-to-use and affordable IVD, performing cancer risk assessment, potentially requiring only a few drops of urine, similar to the simplicity of a pregnancy test. We focused on breast and prostate cancers, as they are the #1 diagnosed cancers for women and men in Europe. This year, these cancers are expected to kill about 35,000 women and 38,000 men. These terrible numbers have increased every year in the last 4 years, at a faster rate than any other cancer worldwide. Like many other cancers, they are very treatable when detected early, but a late diagnosis results in a dramatic decrease of the 5-year survival rate. How can we detect these cancers early? Well, we do screening. An effective screening test should ideally possess 5 key qualities: exceptional accuracy, clear results guiding treatment decisions, easy access for patients, non-invasiveness, and affordability. Such a test not only benefits individuals but also contributes to huge cost savings by reducing unnecessary follow-up diagnostics and treatments in the healthcare system. Now, there is a big problem with the currently available screening tests. They are expensive, invasive, not readily accessible, or completely inaccurate. In Italy, for instance, routine mammograms in public healthcare can have waiting times of up to a year, while a PSA exam is totally inaccurate. These hurdles hinder timely detection and treatment. Moreover, cancer is a major public health and economic issue. Based on projected population aging and growth, the global burden of cancer is estimated to increase by more than 60% by 2040, with over 29 million cases, with an exponential increase in oncology spending. But the great news is that now we have the opportunity to develop a better test and reduce cancer-related costs. We can do that by using a valuable diagnostic asset, the urinary volatilome. What is the trick here? Well, each disease alters the physiological and metabolic status of an individual, leading to characteristic changes in the concentrations of volatile metabolites and the emergence of new ones. The issue, and the reason why nobody has done this before, is that relative concentrations of VOCs to screen cancer are extremely small in the nanomolar range. The trick is that these volatile changes in urine samples can be detected by animals with a keen sense of smell. We hijacked the amazing capabilities of C. elegans, a small nematode. Able to sense nanomolar changes in VOCs concentrations, its olfactory system can distinguish between people with cancer and controls. It is no surprise that a few companies in Asia and the US sell C. elegans-based cancer screening tests, with Hirotsu Bioscience in Japan securing $21 million in funding as a frontrunner. But here we're charting a different course for a different test, which is easier and more accurate. Indeed, already in 2019, we identified olfactory neurons sensing cancer samples, establishing a groundbreaking method for discrimination. By 2020, we elevated our test accuracy to a remarkable 97%. The following year, using sophisticated bioengineering and machine learning techniques, we identified the olfactory receptors on these neurons responsible for cancer detection. And in 2022, we started expressing these receptors on yeast cells, paving the way for an intelligent screening system that aligns with standard screening formats. The key idea of our biosensor is using yeast cell to express C. elegans receptors. Yeast cells possess a single receptor, the pheromone receptor, which we've easily replaced with C. elegans receptors in a safe harbor, a gene cassette that we've created. Then, we've incorporated a fluorescent and a colorimetric reporter in the pathway as a readout. When a tumor metabolite binds to a C. elegans receptors, it triggers an increase in the readout signal. To confirm our genomic machinery's performance, we reinserted the native receptor into the gene cassette and tested it with native agonists. The receptor showed a sensitivity of approximately 1 nanomolar, matching its natural state. To enhance accuracy, we've created 20 transgenic yeast strains, each featuring distinct C. elegans olfactory receptors tailored to detect specific cancer-related compounds. Since each type of cancer possesses its unique metabolite profile, a distinct set of olfactory receptors will be activated. Our next step involves obtaining dose-response curves for each strain to determine sensitivity and screening them with 2 cohorts of 40 samples each, control versus cancer. The system works, and the proof of principle has been demonstrated, but we have to speed up the process to obtain the device. The final device will have 6 features. It will be easy to use with just a urine dip, cost-effective for industrial-scale production using low-cost and eco-friendly materials, highly accurate with over 90% sensitivity and specificity, non-invasive because just a urine sample is needed, and it will detect early stages, providing fast results. Plus, it's portable and requires no special storage arrangements. It has all the features to occupy a competitive market segment. The device will be a simple disposable lateral flow test. It produces a signal upon sample contact, and depending on the intensity of the signal obtained, we may develop a patient-operated point-of-care test similar to a pregnancy test, possibly requiring the use of a plate reader. Eucrasyc will report the level of breast or prostate cancer risk. A positive result indicates high oncologic risk and suggests follow-up diagnostics. It reduces invasive testing frequency for negative results and broadens oncologic risk monitoring to previously excluded population segments. To accelerate market entry, we've pinpointed 4 bottlenecks: staffing and workspace limitations, automation needs, and regulatory hurdles. An additional investment of 3 million euros on top of what has already been internally invested could shorten our industrial research timeline from 3 years to 1 by allowing us to parallelize the R&D processes. Here, the development pipeline, TRL number 5, is ongoing. This means that we are performing the first tests to characterize the accuracy of our system. Furthermore, the 3 million euro investment would enable us to complete our R&D within 18 months. Starting from month 18 to month 24, we plan to begin testing our in vitro device system in collaboration with our partner hospitals, IFO and Gemelli, both in Rome. Concurrently, from month 12 to month 24, we will initiate the regulatory pathway. As a Class C IVD, obtaining CE marking typically requires approximately 1 year of testing, with an estimated cost of 500,000 euros. Let's now take a closer look at the competitive landscape in the field of cancer risk assessment. Currently, the existing tests for breast and prostate cancer primarily fall into 2 categories: immunoassay and molecular diagnostic tests. However, when we look at our direct competitors, we find 2 notable players in this space. The first is Hirotsu Bioscience, which also utilizes nematodes for their testing, but relies on behavioral assays that require specialized personnel and tend to take longer while being less accurate and not portable. The second competitor is PPB Technology, which, like us, employs odorant receptors for detecting volatile molecules. However, it's worth noting that they currently focus on applications related to food testing and have not yet ventured into the realm of tumor diagnostics. In summary, while there are several promising developments in the cancer risk assessment field, our approach, which combines the precision of odorant receptors from C. elegans with urine-based VOC analysis distinguishes us from our competitors, positioning us as a formidable contender in this space. Our roadmap to success unfolds in this 5-year forecast. By the 5th year, with an annual production of 100,000 tests priced at €50 each, we're looking at €9 million opportunity, solely within the realm of active surveillance in Italy. And this is just the initial horizon. Expanding our market across Europe will significantly boost these figures. The peak cash demand occurs between the 1st and 2nd year, and then the company rapidly becomes self-sustaining. We delve here into our SWOT analysis. One of our key strengths lies in our IP protection. We have secured intellectual property rights with 3 patents, and our agreement with Hirotsu Bioscience ensures that there is no limit to our freedom to operate. Another strength is the low production costs. Our product leverages cost-effective yeast-based technology and paper-based support, which significantly lowers production expenses. While we're confident in our technology, the challenges we face is introducing an innovative product, especially in healthcare. Navigating regulatory pathways and achieving widespread adoption can be complex and time-consuming. On the bright side, the opportunities are vast. Launching our device would place us at the forefront of the diagnostic industry. Success could make us industry leaders, revolutionize mass screening, and significantly impact public health. However, it's important to acknowledge potential threats. Although currently lacking, we may face competition from industry giants with more resources and advanced technology. Staying ahead of these competitors will be a challenge that we need to navigate strategically. Our scientific team consists of 7 researchers with diverse skill sets and backgrounds. This multidisciplinary approach has played a crucial role in our progress, enabling us to leverage a broad spectrum of knowledge and expertise to address various challenges in our field. Our collaborators include Sapienza University, the Italian Health Institute, and several hospitals. The engineering team is responsible for the design and development of the paper-based prototype and reading platform that will then be distributed to diagnostic centers. This slide showcases our board members, each with specific roles and extensive industry experience guiding our company to success.